Welcome everyone to our instructional video on genomic DNA library preps. We're using the SparQ DNA Frag and library prep kit, but there are other kits you could use, like New England Biolabs makes one called, I think it's NEB Next Ultra DNA kit. So it's very similar to the one we use. So there are substitutes, but we use this one because that's what's being used in our genomics core. So I'm Craig Barrett. You all know me. I'm an assistant professor at West Virginia University, plant evolutionary genomics. To my left, or to my right, is Hannah. Do you want to go next?
Hi, I'm Hannah Thixton. I'm a PhD candidate, obviously in Craig's lab. I study orchids and their interactions with their fungal hosts.
Hi, I'm Sam Skibicki. I'm a PhD candidate as well in Craig's lab and work on Asteraceae, and I use genomic libraries all the time.
Hi, I'm Cameron Corbett, and I'm a PhD student in Craig's lab, obviously, and I study invasive Japanese stoatgrass.
Yeah, and Cameron is the one we filmed doing this whole thing, so you'll see him in the picture. So I'm going to share my screen, and we'll get the presentation started here. Okay. Well, now okay. Okay, so just a little brief intro here. So before you begin your library preps, what you're going to want to do is quantify your genomic DNA sample concentrations with a qubit broad range double-stranded DNA kit. That's that first line there. So that's going to be important. We may not do it for this workshop just because we want to go through this process once, but that's going to be a thing you want to do. And then what we typically do is dilute our samples to 20 nanograms per microliter. So we try to get a standard concentration across all of our samples. If they're below 20 nanograms per microliter, which sometimes they are, sometimes you're dealing with difficult material or barium material that doesn't give very large yields, then you can use extra input to DNA to get up to about an equal amount of input DNA as you would for a standard prep. We always run gels to visualize our genomic DNA, especially with our barium samples. So in that way we can assess the level of degradation. And I'll show you a gel here in a second that illustrates that. And typically what we do is based on whether we're dealing with herbarium samples or fresh or silica dried samples, we bin these into two categories. So the first would be degraded DNA, so with evidence of degradation, having no visible high molecular weight DNA. And then the second category would be non-degraded DNA. Okay, so in this case, you'd have some or all of the DNA being high molecular weight. And to show you some examples here, on the left, you'll see some high molecular weight genomic DNAs, which these are palm leaves that I flash froze in liquid nitrogen and then got them immediately into the -80 freezer. And so the thing that varies the most in this protocol is the fragmentase time. So you're going to use an enzyme to basically chew up genomic DNA into smaller pieces that can be sequenced. For high molecular weight genomic DNA, you're going to have to do that a lot more because you're approaching like whole chromosomes essentially. And so this is what high molecular weight genomic DNA looks like. You see this big thick band here, larger than 20 kilobases. That can be actually much larger. And then maybe a tailing of smaller fragments. But this is what high quality genomic DNA looks like. If you're extracting DNA from herbarium records, it's going to look something more like this. And so you're going to see a variety of different levels of degradation depending on how old the specimen is, how it was preserved, what conditions it's been in a particular herbarium, et cetera. And typically what you see is a smear of low molecular weight DNA. Here, this ranges from 100 to 500 bases, but then you have this tail of higher molecular weight DNA. And the sweet spot here is going to be around 500 bases. That's what we want our target libraries to be for optimal Illumina sequencing. And you can see that all of these samples have some DNA in that range. And so we're going to do library preps on this. And you can think of herbarium DNAs as like pre-sheared DNAs. So you don't have to do that much fragmentase. In fact, some people don't even do a fragmentase step at all, or they don't sonicate, for example. The reason we do a 1 minute fragmentase time is so we can prepare the fragments for adapter ligation, basically. So we do it for 1 minute as opposed to 14 minutes for high molecular weight DNA. Now, some of you will be doing ISSR-seq as part of your projects. And so these are ISSR amplicons that were run out in a gel. And typically we see these bands between, I would say, like 200 bases and then several KB. And so we've optimized this at a fragmentase time of about 3.5 minutes, which seems to work really well for targets in that range. Okay, so we use the Quantabio SparQ DNA frag and library prep kit, which is really an easy, straightforward kit. You used to have to do every single step with a cleanup back in the day, and it took you a whole week to do library preps, but now we can do it basically in a day. Okay, so the first step we're gonna talk about is the fragmentation and polishing. So we're gonna chop up the DNA and then fix the ends of the fragments. So here we're gonna start our little video. There's Cameron in the lab. Maybe he can explain what he's doing.
Yeah, so this step is the frag and polishing step where we're basically breaking up the DNA. So in this step, we first add the buffer, H2O, and enhancer solution to a master mix. And then I'll take that master mix and divide it up into each of the PCR tubes. And from that, I'll add the required amount of DNA that's been either diluted or combined to get a roughly 20 nanograms. And then at the very end, I pull the enzyme out of the freezer and add that the last step before putting it into the thermocycler.
So I was the video editor for all of these videos, and I try to put in some tips. So with these steps, I highly recommend preparing everything on ice. If you happen to have a cold block, that would be even better. When you're adding your buffer, your water, and your enhancing solution, feel free to take your time with this because this is not a time-sensitive step. The time-sensitive step, which will be shown in a video in a little bit, is when we add the enzyme itself, the fragmentase. And that's the one that is very time-sensitive.
So why is it time-sensitive?
The reason it's time sensitive is because the second you add that fragmentase, it already starts to break down the DNA. So the most reactions that I'm able to do at a given time is 36, which is kind of pushing it for a lot of reactions. I have a way to put the fragmentase on the wall of the tubes. That way I can get all of them added in. I spin them down and immediately put them into the pre-chilled thermocycler. And that was basically my limit. That's the fastest I can go, but I would only recommend, like, especially when you're first starting libraries, just do 4 reactions, just so you can kind of get a feel of when you add the fragmentase. This is the only step in the whole library protocol that is very time sensitive.
Right, so the issue is, like, the second you add the fragmentase to the first tube, the time between that first tube and the last tube, if you're doing 36 reactions or something, is going to be the time it takes to individually pipette 36 tubes, and that's a few minutes. So yeah, we start this protocol only doing a few samples, work your way to more samples as you get more comfortable with it.
Mm-hmm. I do have a tip for the pipetting mix without making bubbles. I actually learned it from Ryan Percifield. He runs our genomics core here at WVU. You basically I'm going to use my coffee as an example. You take your tip and you have the solution, you aspirate it in and out like this. And that prevents it. 1, you get a perfect mix. 2, you prevent bubbles from happening. I know the one time I first did this, I had bubbles in my reaction. And I had low quality library products in the end because of the bubbles. So just a heads up so you guys don't go through what I did.
So what step are we on now, Cameron?
It looks like all the reagents are in the tubes now. And I was making sure there was no sidewall on there before they go in. So everything was set up, so this must be the enzyme being added, the final step.
Okay, here's the critical step, adding the fragmentase.
It's important to go as quickly as you can, but still be careful and just make sure don't get bubbles, don't mess up. Just be cool and calm and work.
Cool and calm. I know a lot of people will be doing this for their first time, so don't worry if you screw something up. It's not a big deal. This is just practice. So.
And if you're only doing 4A, you don't have to speed through it so fast. Just move at your normal pace. It'll be fine.
I do like the suggestion of sticking the fragmentase blob to the wall of the tube, so it's not in the main reaction. And are you adding your DNAs last, or is that?
Everything's in.
Okay.
The last thing I add is the fragmentase.
Okay.
And that's why I try to have it stick on that sidewall.
Yep.
Then I make sure there's nothing sticking to the wall of the plate. I spin it down. Yep. Immediately goes into the thermocycler. And be sure that your thermocycler, like, this is a great time to actually have your thermocycler running before you start setting up your reactions. If I remember correctly, for SparQ, most of the reagents are liquid. I don't think there's many liquids at this step that are frozen that you have to wait to thaw. So just have the thermocycler at the minus, I think what we have at 4 Celsius. Just have it running in the background. That way it's ready to go for your next steps in the thermocycler.
We're going to show the thermal cycle here in a second. It's pretty easy. It's just a single temperature, and then you raise the temperature and kill the enzyme activity. Should we go to the next slide yet, or?
Yeah, I made this video where it's like you can actually see the full time it took Cam to do 8 reactions, but we don't have to watch the whole thing.
Okay, well here we go. We're about to.
Oh, there it is.
Okay. This is the fragmentase step. Okay. There's the fragmentase.
I think the next clip is the thermocycler.
Okay, well, I'll skip to that here. And
so I can talk a bit about the thermocycler. So we started off at 4C, like we mentioned earlier. That's just to keep things cold so the fragmentase isn't chugging along and chewing up things faster than we'd like. Then the second step is going to vary between what you're doing, whether you have really high quality DNA and you're chewing for 14 minutes of fragmentase time, or 1 minute for degraded herbarium DNA, or even something towards the middle, like 3.5 minutes for the ISSR protocol. Then when you bring it up to 65C after after all that time at 32 is when you're killing the enzyme, and then you bring it back to 4 when things are finished up.
Yeah, so a pretty simple protocol, and the only variable is the amount of this 32C fragmentase time, which is the optimal temperature for that enzyme to be active. So it looks like we're going over to the cycler here. And there's just a shot of the protocol again for you. You can feel free to pause the video at any time as you're doing this and just follow along. And so now we're on the thermocycler and the end of the video. So we can move on to the next step. So the important thing here is at this point, after you've run the PCR cycle, you need to proceed immediately to adapter ligation, which is the second step here. Okay, so the arrow is pointing now here to the adapter ligation. So you have your fragmented genomic DNA, hopefully mostly in the range of about 500 base pairs. And now what you need to do is stick your adapters on. And those are going to be the things that allow you to actually do your sequencing. Okay, so that's what Cameron's doing right now. Here's our reaction setup. Somebody want to give a little intro here?
Sure.
Yes.
Oh, I'll go, Cam, since you already talked the last one. So I highly recommend preparing this adapter ligation mix in the last 10 minutes of the previous thermocycler. So when you're that last 10 minutes of the 64 C, I can't remember the exact temperature. But what you're doing, 65, I was close, off by 1. But what you're doing with the adapter ligation is now we need to actually add on the Y adapter to our broken up chunks of DNA. So in order to do that, we have the ligation buffer, we have DNA ligase, which is going to ligate on our Y adapters, obviously nuclease-free water, and your total volume you'll be adding to that previous product is 18 microliters. What's nice about this thermocycler step is it's only one step. It's at 20 degrees Celsius for 25 minutes just to ligate on those Y adapters. But be sure that you have the heated lid setting off. Otherwise, you will roast your DNA ligase, which what we did when we were optimizing this protocol. It was on accident, and it just roasted our products. We had nothing to look at.
So you'll want to set up all of your thermal cycles before you ever start doing anything with on the bench. So you want to make sure they're all set and you can find them pretty quickly, and then so you go right into this step directly with the product of the fragmentase reaction. So there's nothing in between. You just add this right to that. Correct? Okay. So there's the more detailed protocol. Oh, actually, I think this is actually blocking the. Okay, there we go.
But yeah, so what was not mentioned in the master mix was that Y adapter. So you add the Y adapter to each tube. You don't add it to the master mix. You add the Y adapter to each tube individually. Then you can add your master mix, pipette mix, of course, about 5 times. Don't make bubbles. And then you just proceed immediately back into the thermocycler. Is there anything else that we need to add here?
I mean, if you get some bubbles or spin it down real quick, make sure there's nothing on the sides. Just make sure it's all in one nice pool in the bottom of the tube.
And if I have bubbles, sometimes I'll take a clean pipette tip and just pop it.
Yeah, sometimes spinning down might help too. Mm-hmm. One thing we should talk about with enzymes is, in general, don't vigorously vortex an enzyme because you can destroy it. So you can spin it down, that's fine, but like pipetting up and down is generally a better way for mixing these reactions than vortexing. If you have an enzyme in the mix, if it doesn't have an enzyme, you can vortex just to mix things up in your master mix, but otherwise refrain from doing that. So we ready to?
Yeah.
I keep photobombing Cameron here in this video. So okay, so you've put your adapter ligation reactions on the block for how long is it? 20 minutes, 25 minutes of 20C, and now they're ready. Okay, so you take them directly off and you proceed to your first bead cleanup. And so you should all have the magnetic beads and hopefully a magnet to use. And does somebody want to kind of briefly take us through the cleanup protocol we're using?
Sure, I'll go again. So with the bead cleanup, what I like to do is right when I put the adapter products into the thermocycler for that 25 minutes, I immediately pull out the beads so they can just have that time to get to room temperature. Sometimes I hold it in my hands just to kind of speed up the process of getting these beads to room temperature. It is important that you have these at room temperature, and make sure that you vigorously vortex these beads before you add them to your products that you're cleaning. After you vortex your beads, you'll add 1.3 times the volume of the reaction, or 32 microliters to each sample, and you'll just pipette mix. I usually do 3 to 5 times. After your last sample, I like to set a timer for 5 minutes, mainly because I will forget, because we're size selecting for the right range. So about that 300 to 500 base pair range for our products that we want to continue on with the next steps with the library amplification. After those 5 minutes have elapsed, you'll pellet the beads using the plate, which if you look at the bottom left picture, you can see that the beads on the magnet are pooling. So what you're looking for is a clear solution at the end. You'll remove that supernatant, you'll remove it from the magnetic plate, and you'll wash it with fresh 80% ethanol. It is important that you're making fresh 80% ethanol every time you're doing library preps, because 80% ethanol, it's a high percentage, it'll evaporate over time. I always just make a very small volume of it, just enough for me to do my library preps, and that is it.
Yeah, if you make your ethanol, you can do it in like a 50 mil Falcon tube, and that's usually plenty for cleanups, like for an entire week.
Yep, that's what I do. I put in a Falcon tube and I just call it a day. You'll repeat that ethanol rinse, like you'll pipette mix your ethanol to your beads, so that way they're resuspended again. You'll put them back on the magnet, discard that supernatant, and you'll repeat it for a total of 2 rinses. Then on that final, once you aspirate that final solution, you'll place the beads to air dry for about 5 to 10 minutes. This is probably the most time-consuming spot when it comes to library cleanups for the adapter ligation and the library amplification cleanups. The beads do take some time to dry, and some of them will dry unevenly. So what we've started to do is, if there's some beads that are starting to dry and are starting to crack, we'll have those beads, we'll add some of that Tris HCl that we'll be providing you guys to the beads, so that way they don't crack. If they start cracking, it's just gonna make it harder and harder to elute these beads back into solution. Once you resuspend your beads, just let it sit for 2 minutes just to kind of rehydrate those beads again. You'll pipette mix, put it back on that magnet again, and you'll transfer only 7.6 microliters of that Tris solution to a new PCR tube. This is where you can actually stop. So this is a breaking point. So you can either store your samples at -20 and come back the next day. That's what we like to do in our lab, is we like to split this protocol into 2 days. Or you can continue on from here. So the choice is yours. It usually takes about 2 to 3 hours for the first 2 steps, including this cleanup.
So you could do it in a full day, right?
Yes,
like you could
absolutely.
do the whole thing in a full day and then run the gel at the end. So just to reiterate here, step number 10. So that last step, when you're putting the beads in solution back on the magnet, and you're pulling off that 7.6 microliters, that's the part that has your DNA in it. That's the part that has your adapter ligation products in it. So you're going to keep that, right? So you've discarded the liquid up to now, but now you're going to keep it, because when you put it when you put the beads back in tris HCl, the DNA fragments will dissociate from the beads and they'll be in solution again. And so you want to capture that, and then you can throw out the beads, or keep them if you're really paranoid. Okay, so there's our cleanup protocol. Get used to this. You'll do it a bunch of times, depending on what protocol you're doing, with only slight variations. But once you go through it a few times, it's pretty straightforward. Okay. Now to our fourth step, and that is, so we've got our adapters ligated onto our library fragments. Now what we need to do is what's called our indexing PCR. So you're going to do a whole library amplification, but you're going to do it with specific sets of primers. And we use dual indexing. So your 5' primer is going to have, are they 8-letter or 6-letter barcodes? I can't remember.
It should be 6 for the 5'.
Okay, so you'll have these unique barcodes, one on the 5' primer and another on the 3' primer. We call those what I7 and I5 primers. It's just the protocol we use. And so basically those primers will have so the adapters will have tails. Those primers will match the adapter and then have a unique barcode on them. So when you do this PCR, you're basically tagging each one of your samples with a specific forward and reverse barcode, right? And so the dual indexing comes in by, you don't just have a unique barcode on the 5' side, you have one on the 5' and on the 3'. So if you have 8 forward primers and 12 reverse primers, that's 8 times 12 combinations, right? So you can have, that's 96, right? I think math is hard. Okay, so anyway, here's the PCR that Cameron's setting up. Somebody want to go through this? I'll skip to the reaction setup and thermal cycle.
Yeah, so this is a pretty simple to do step. You just add 10 microliters of the pre-mix master mix straight into each tube that you pull. We usually pull and have thawed from the freezer, but it can just be straight from that last step, the cleanup step, if you are doing it all in one day. And then you go through and add 1.5 of each of the primers. And so each tube is going to get a unique set of primers. So there will be no overlap or no identical primers in each tube. That can be a little time consuming. You just want to make sure you get it all right, because you won't know until it's too late if you mess up. But after that, you just put it in the thermocycler and run the settings that are shown here on the left, and it's pretty simple and straightforward.
And so I think the important part here is the when you're running your cycle, you can vary the number of cycles. And so if you do too few cycles, you're not going to have enough product. If you do too many, then you run into another set of problems, where if you do too many cycles of PCR, you start to get chimeric PCR products. You start to get PCR duplicates, which are exact copies of the same fragment instead of unique copies. And these are things that basically are going to cause issues downstream. So there is a sweet spot in terms of the number of cycles. You don't want to do too few, and you don't want to do too many.
Yeah, so we typically do 12, which is kind of generally assumed to be on the higher end of what you want to do, but it's the sweet spot that works for us.
Yeah, and if you find that you're getting way too much product, then you can tone that down to like 11 or 10. If you're getting too little, you can bump it up to, how high do they recommend even going in terms of the number of cycles?
I think like 10.
10? Okay. I've done it up to 18 before for some really, really wacky libraries, but that was a different protocol. So yeah, 12 seems to be our sweet spot, but you could always adjust this later if you're not happy with the results. Okay, so should we skip ahead here? So this is the step you're making more copies of your library, and also tagging it with unique 5' and 3' barcodes. Okay, so here's the more detailed protocol. And it's a master mix, so it's pretty simple to set up. The most time-consuming part is adding the primers, right? The unique primers.
Mm-hmm. What I would recommend to keep track of all your barcodes is to have it like a master Excel sheet where it has your, not only your sample ID, but what barcode they got, like which I5 and which I7 barcode they got. So just to make it much easier for you later on when you're actually doing data analysis.
Yep, and we'll provide the spreadsheet with the unique primers on it, and then you can use that to keep track of your samples. I'm trying to remember the kits that we sent out to the regional PIs. Did we include unique barcodes, or did we just include one set of primers?
I think
one set.
Okay, so you won't actually be using unique primers for this protocol, because it'll just make things a little easier. But in the future, when you start doing this, you'll want to add a unique forward and reverse primer to each sample. And if you think of this setup in terms of like a 96 well plate or a bunch of tubes in a rectangular setup, you can have your 5' primers on the vertical and then your 3' primers on the horizontal, right? So think of it like a grid. Okay, so then we go pretty much directly into a cleanup. I would recommend just doing this right after your reaction ends, and then you get to another stopping point. And the only difference between the last cleanup and this one, well, there are two main differences. Here you're only using 20 microliters of the beads, a smaller volume. That's a 1-to-1 ratio. Everything else is going to be the same. I think you only do one ethanol cleanup here, right? So it'll go a little quicker. And then after, you elute with 19.2 microliters of tris HCl. And at that point, once you've eluted your amplified libraries, you've reached a stopping point, and you can put these in the -20 C. So this is basically your second stopping point, right? So the first is after the adapter ligation cleanup, and the second is after the whole library amplification. So there are 2 stopping points here before you go on. Okay, so there's your second cleanup. And the next step is going to be QC, so quality control and quantification of your libraries. So somebody want to take us through this bit?
So we use qubit to get our DNA quality or quantity. We use a gel for the quality of it. So for quantity, we just use the high sensitivity for libraries as opposed to the broad range. So we create our qubit solution, which is just a mix of the solution and then the dye. And then we add 190 of that plus 10 of the standards for the 2 standards. And then for our actual DNAs, we do 198 of the qubit solution and 2 microliters of our DNAs. And we do that for each one, make sure to vortex them real good, mix them up. And then we go to the qubit and run them, and that will give us our DNA quantity. And then we always run it on a gel too, just to kind of visualize how the quality of the DNA. And that's what the gel image on the left is. And you can kind of see where the brightest part of the band lies is kind of roughly like how big the fragments of our libraries are. And we're shooting for around that 500 base pair size.
Right, and so these, you can see there's like a tail of larger fragments here, but in the last step, we're gonna do size selection. And that's done at the genomics course. So we're gonna select a really thin band of our think of these as like fragments that represent random bits of the genome, right? You've chewed it up, you've prepped your libraries, and then we're gonna select a really thin band right around 500 bases, more or less. Usually a little less than that. And so here are the results of a qubit, a really nice qubit that Cameron did with some really really high concentrations here. Maybe some of them are a little too high, so you can dilute them later. We'll talk about that in the next step here. And just a note on the high sensitivity versus broad range qubit kits. So there are two different chemistries you can use. One is more sensitive to the other. So the broad range kit, I think, can detect up to 1,000 nanograms per microliter. And the high sensitivity kit only goes up to 100 nanograms per microliter, but it's more sensitive at the lower end. And that's why we want to use that for library quantification, because usually these numbers are, I think, what's our target, between 3 and 30 nanograms per microliter?
Yeah.
For library.
Yep, that's what we aim for.
Libraries. And so that kind of brings us to our next question. Well, what if I go through this and I do the library amplification, but it's still really low, like below 3 nanograms per microliter? So that's really kind of on the low end of what you'd want to take forward into sequencing. And it makes pooling more difficult if you're below 3 nanograms per microliter or above 30, because you have to get them pooled at equimolar ratios. So luckily, if it's too low, you can re-amplify it again using another kit. And so somebody want to take us through this bit here?
So with the kappa kit, we didn't send any kappa kit, did we?
I don't think we did.
Well, if there are samples that are too low, I think we'll probably come in and help you guys out with this, with the kappa kit. This is a very straightforward reaction. So what you'll do is you'll have to, after from the qubit, say if you have anything that's less than 3 nanograms per microliter, but they still showed up pretty well on your gel image, that's where you'll decide, like, okay, let's re-amplify these library products to kind of salvage, like, that's what I would say, to salvage this product, so we have to go through that whole process again. Do not use the kappa kit if your concentrations are very high. You'll just have to redo your libraries, but for those that are too low, we can use this kappa kit to re-amplify those products, so that way they're at that 3 nanogram per microliter range. Something special to note is we're actually using the primer mix that's from the SparQ protocol and not from the kappa protocol. So yeah, Craig's kind of highlighting it. So make sure when we're using this, we use the primer mix from the SparQ kit. It is not meant to be for building libraries. It's just to help re-amplify products. So we'll take 0.75 microliters of that primer mix with the kappa master mix, and then we'll use as much of the adapter ligated DNA as possible. If you don't have that much product, because it is very common, especially after you run a gel and after you qubit, you may not have as much DNA product. You'll just adjust your DNA concentration. You'll just add more water just to bring it up to volume. And then you'll use these following thermocyclic conditions. I found that 12 to 15 was kind of my sweet spot when I did my kappa re-amplifications. But I was also using high quality DNA. I'm not so sure about herbarium samples, however. So maybe Cam or Sam can pop in on that.
You're talking about number of cycles, right?
Yes, the number of cycles.
Okay, so the 10 to 18. So you found that like 12 to 13 or 12 to 15, you said, okay.
Yep.
That's probably good.
It also really depends on your incoming concentration. Sometimes it's maybe 2.9, it's just below that 3 nanograms per microliter threshold. So you're going to be on the lower end, closer to 10 cycles of re-amplification. But if you have something that maybe even the high sensitivity qubit can't even pick up, then you might be more towards the higher end.
So what's the lowest concentration of libraries going into this re-amplification that you've successfully saved, or that's, you're kind of rescuing a low concentration library here, right? Somebody.
So when you're below, I think it's 0.5 nanograms per microliter, even though it says the high sensitivity qubit only goes down to 1 for sensitivity, it can detect things to 0.5. But if it's below that, it says too low a concentration on the machine. And I have pulled ones that were that low back into concentrations that are usable. So it's hard to tell how low they were to start, but they're below the detectable range and still savable.
Yeah, there's still maybe a little bit of library you can salvage there. But again, you want to be careful and not do too many cycles for something that's already high, because then you're introducing chimeras and and PCR duplicates, which will have to be removed later on because they're not really adding anything unique to your genomic library. They're just copying fragments that are already in high abundance. So yeah, this is kind of like if you go through the protocol and you still have a low concentration of your library, you can use this Kappa library amplification kit, which is specifically designed just for that. But we're using the primers from the SparQ kit that match our adapters basically. And this is just a general primer. It doesn't have a barcode on it or anything. You've already barcoded your fragments, and you're just going to amplify all of those fragments and individual reactions to bring them up to concentration. And of course, you'd have to do another cleanup after this. That's, you know, anytime you mix buffers and primers in, you're going to have to do a cleanup. So there's one last cleanup. Do we do just one ethanol wash here? And we use 25 microliters of the beads based on the volume. Okay, and one ethanol cleanup, but everything else is the same. Okay, so there's your final cleanup. So for your more difficult libraries, you're gonna have to do 3 cleanups, and you're usually gonna have a couple that are kind of problematic that you have to fight with a little bit, but you can get them up to the right concentration for pooling, which is our next step here. And this is probably the last step you may be doing in your labs is library QC. So you're gonna want to cube it and then probably run a gel again. And you can run a gel with as little as 1 microliter and still see your library. And so if you're dealing with small volumes and you don't want to use a lot of it, you can use 1 microliter of your library to run on a gel, and you can still see it usually if it's detectable. And then you're going to want to do the qubit high sensitivity double-stranded DNA assay again to see if your library is in that 3 to 30 nanograms per microliter range. That's your target range, and that's going to make pooling a lot easier. Do you want to talk about pooling? Anybody? Your experience with pooling?
So my experience with pooling, I always get outside advice just to kind of confirm how I'm going to bin my pools. So I usually break my pools into 2. I know Sam has done 3 for his herbarium samples, but what I try to aim for is a low input pool versus a high input pool. For example, I'll pool together my 3 to 15 in one pool and then the 15 to 30 in another pool. That way it makes it a lot easier to actually do the math to adjust the volumes as needed per pool. I actually have the equation here where it's basically you just take the qubit value and you just determine what is X. Basically, we're aiming for 10 nanograms per microliter, and then you'll get whatever you need to add of your sample to your given pool. And what's nice is you can keep pools once you pool them in the freezer for a few months. That was based off of my samples. They were in the freezer since January, and they were just shipped off, this month? Last month?
Last month, I think.
Last month, and they worked great. So after you get through the libraries, it's pretty much smooth sailing from there. And
you can store them long-term at -80. So
yes.
So your libraries will stay stable at -80. What was I going to say about pooling? So what Hannah was just describing is instead of just doing a single pool for 96 samples, you can subpool them maybe in sets of 8 or 16 or 24, however many bins of concentration you have. Say you have 24 that are in the 3 to 10 range, 24 that are in the 10 to 20 range, and then another set that are higher concentrations. You can pool them based on common concentrations. It'll make things a lot easier, because then you won't have to be using drastically different volumes and decreasing your accuracy. So you can make say 3 or 4 subpools of your libraries. Then you can qubit those again to get the final concentrations, and then you can pool those together into a single pool. It makes things a lot easier in terms, you might think doing this twice might increase your error, but actually it decreases it because now you have a better idea of what's going into your final pool. And you've quantified your original libraries, and then you've quantified your subpools, and then you make your final pool and you can qubit that again. and figure out what your final pool concentration is. So the goal is to get every sample as close to the same molarity as you can. So the same concentration as you can. And in terms of like what you'd be doing in your labs, if you don't feel comfortable pooling, then you can just send it to us and we can do it for you. Everything after this step is going to be done at the genomics course. So once you've gotten to this point, congratulations, you're pretty much guaranteed to get some really good data. And so the last bit is going to be the stuff that happens in the genomics core, which you don't really need to know too much about, but it's good to know what's happening with your sample. So you have your total pool, which you've quantified, and you have equimolar ratios of your different samples. We'll handle things from here. So what happens in the genomics core? So we handed this off to our staff, Brian Persifield. He's our genomics core guy. Some of you might know him. So what he'll typically do is run your sample on a machine called the bioanalyzer, which is like a really small gel that runs out and quantifies your sample. So you get not only the length distribution of your fragments, but you also get the relative intensity of fragments of each size, right? So this, you use a ladder here to standardize everything. And here we have a pool that peaks at about 350 base pairs, which is pretty much in the range we want to be. So 300 to 500 base pairs is a typical library. Some are shorter, some are a little bit longer, but anything longer than 500 in Illumina has issues with sequencing. So this is kind of the optimal range. Most pools straight from library preps don't look like this. They're kind of more broad and flat. And so what we do in that case is we do some size selection. So we thin that distribution on a machine called the Blue Pippin. So this is another gel-based application that runs your sample out and then cuts out a fragment. So you can do this on your own if you want to on gel, or you can potentially do it with beads, different concentrations of beads. People have done it that way, but the most accurate way is to use this specialized piece of equipment that is specifically used for size selection. And that can get you a nice tight distribution like this, which is what you want to see in your final library. It's going to maximize your sequencing yield and prevent any other problems downstream. And then the last step is to use quantitative PCR to do a final quantification. And then what ends up happening is your final library, you're going to use a very small amount of that for actual sequencing. So I think they dilute it down to like 10 picograms or some really, really small amount of library and use that. So when your library goes off to the sequencer, it's typically a really small diluted amount of it. And we usually keep your libraries in the freezer in case something happens at the sequencing step, we could retry it or something. So you're only using a really small amount of your final library pool for actual sequencing. Well, thanks everybody. I think we'll wrap things up here, and feel free to pause this as you go through the protocol and rewind if you want to see another part again. But use this as a guide to getting you through your first library prep. So I'm going to stop sharing here. I think Cameron disappeared. Probably had an internet issue, but yep. I'm going to stop recording. Thanks, Sam. Thanks, Hannah. Thanks, Cameron, wherever you are. Best of luck to everybody with your libraries. See ya.